Good morning, everyone. I am welcome you all to this 23rd day of CSIR Life Science Question and Answer discussion session. Today we are going to discuss about the Part C questions of Unit 2. Yesterday itself we discussed the Part C questions in Unit 2 because as I mentioned, the 4, 5, 13th units are very important. Other than that, the Unit 1 and 2, it's 1 to 6 sevens are the next level important units. So we will enter into the session of cellular organization Part C questions. In this, one thing is very important. Whenever you are studying about the cell system, just study like a pathway diagrams. Okay, each and every questions are carrying a very important portion because one pathway carries nearly 4-5 questions. Just have it in your mind. Because when you are studying for your CSIR examination, you want to collect a book and find out one topic. Means in that topic you need to draw the pathways like our flow chart, anything. Just draw the flow chart and then go for the study. Okay, we will enter into the session now. In this unit 2 part C question. First question is, in an attempt to study the transport of secretory vessels containing insulin along microtubules in cultured pancreatic cells. Okay, what they did in an attempt to study a transport of secretory vessels containing insulin along the microtubules in cultured pancreatic cells. This is a study. Then here they asked, how would treatment with calcimide, affect the transport of vessels? Here the main key word is the calcimide. This calcimide function, this what it will doing. This calcimide is a mitotic inhibitor. Very important thing. The calcimide, it's the mitotic inhibitor. It can inactivate the spindle fiber formation. So it will inactivate this spindle fiber formation. So that is microtubules are inhibited as per this statement. What is the calcimide? That is the mitotic inhibitor. It can inhibit the inactivate the spindle fibers. Here, so they are prevent the formation of spindle fibers. So the microtubules are their spindle formation is the polymerization process. So as per the statement, the B is the correct answer. It will affect the polymerization of microtubules is inhibited by calcimide, which in turn would inhibit the transport of secretory vessels. First step, it will inhibit the polymerization of microtubule. Due to that, the transport of secretory vessels are inhibited. This is very important concept in the cell cycle. It can also destroy the already formed spindles also can be destroyable by the calcimide. This calcimide affect the transport of vessels because of it's inhibiting the polymerization of microtubules. It's a concept. This question and answer. Next question. Chromatin condensation is driven by protein complex called as the condensin, which are membrane of family of structural maintenance of chromatin protein that play a key role in organization of eukaryotic chromosome. This is the statement. This is a key story. Then condensin along with another family of SMC. SMC means that is structural maintenance of chromatin protein called as cohesin. This cohesin significantly contribute to chromosome segregations during the mitosis. And if the cells are treated with inhibitor of CDK1 phosphorylation immediately before the cells enter into the M phase. When they add the inhibitor, cells are enter into after the a M phase. Okay, the cells are enter into the M phase. During that time, they are adding the inhibitor. What inhibitor? That is CDK1 phosphorylation. Here, what is the question? Which of the following statements most likely true? Okay, in this as per this schedule, this slide, you can understand what what the way we want to study the mitosis and meiosis, because mitosis and meiosis, we are all a very important concept in cellular organization. In that mitotic process and meiosis process having n number of steps, so only I'm told you need to draw the pathways and you need to draw the structures. You need to each and every flow chart, you need to draw the flow chart for this mitosis, meiosis and their protein pathways. So as per their statement, what the chromatin condensation driven by the complex called as condensin, these membrane proteins are that play a key role in the organization of eukaryotic chromosome. This is the statement. Then condensin along with another family of protein that is called as cohesin, it significantly contributed to the chromosome segregation in the mitosis. If the cells are treated with the inhibitor with CDK1 phosphorylation, so here you want to know the role of the CDK phosphorylation, then only you can answer this question. So here answer is sister chromatid are held together by the cohesin along with the entire length of the chromosome. The answer is sister chromatids. Okay, this sister chromatids means in they are playing, they are look, they refer identical copy of the chromatid. Already it's look like already available chromatins. It's normally formed by the DNA replication process, and that duplicate, that exact copy of the cell production held by the cohesin along with the entire length of full chromosome. That is not happen with the condensin. The sister chromatin formation is held with the help of cohesin. Okay, it's a very very important thing. So cohesin is the very key compound in this question. Okay, in this one more compound, this cohesin is a complex protein. Okay, this complex protein is the only reason for production of the homologous recombination, that is sister chromatid production. Okay, so this is very important. It's conserved in the eukaryotic, highly conserved in the eukaryotic. Next, Rec8 is the meiosis specific cohesin. It's one specific compound, that is the Rec18. Rec8 is Rec8 is a meiosis specific cohesin, in that maintained a chromatic cohesive between the sister chromatid in a meiosis 1. Which of the following phenotype listed below would be the predict will be main first in Rec8 yeast? Okay, they are doing a that process. They are doing a research the recombination with the this yeast cells. So in that study, they are getting what kind of result, or that is a question. So Rec8 is meiosis specific cohesin. That is the statement that maintain the chromatic cohesive between the sister chromatids in meiosis 1. This is the statement. Phenotypic listed below, that here they are given a phenotypes that you want to answer. Select the answer. It below would be predict will be manifest in Rec8 yeast. So 8 answer is B, the improper reduction, division and low viability of T-threads. This is the answer. Then here next question, when circular plasmid having a centromere sequence are transformed into yeast cell, they replicate and segregate in each cell division. Okay. However, in a linear chromosome is generated by cutting the plasmid at a single site with the restriction endonuclease. This is the whole story. Next, the keyword, the next statement, the plasmids are quickly lost from yeast. And here next is a key sentence. It is known that gene on the plasmids are lost because of the instability of chromosome N. This is the key. What would done so as to restore its stability and can inherited. So in this process they are provided the story and ask the question from the story. So first we want to understand circular plasmids having the centromere sequence are transformed into the yeast cell. They are replicated and segregated under cell division. So they are transformed into yeast cell. In that the cell division happen. However, if linear chromosome is generated by cutting a plasmid at a single restriction site. So circle is there. We want to get a linear means we want to use the endonuclease enzyme. So that is the process. The plasmids are quickly lost in the yeast. It's disappear in the yeast. It is known that in plasmid lost because of the instability of the chromosome. And that what could be the restore stability can inherited means the answer is by incorporating telomere sequence to the end of the plasmid. So this telomere sequence incorporation that would improve the stability. Next question, glycolipids and sphingomyelins. This if you are well versed in biochemistry, you may understand this concept. This glycolipid and sphingomycin myelin are produced by addition of sugars or phosphoryl choline to ceramide on the cytosolic and luminal surface. That respectively they are added for of the Golgi apparatus. Finally, after such a modification, these molecules are located on the outer half of plasma membrane. This is the process. After this, that phosphorus addition of sugar. Then, what key event is responsible for such a localization? What is the localization process means? This that are moving in towards the internal or external part. The glycolipids and sphingomyelins are produced by the addition of sugars or phosphoryl choline to ceramide on the cytosolic and luminal surface respectively. So in this process they happen in the Golgi apparatus. Finally, after such a modification, these molecules are located on the outer half of plasma membrane. What is the key events responsible for means that is the action of B answer action of flippase and membrane fusion. Okay, next is just a trickiest question but logical question. When one isolates ribosome from bacterial life state apart from 70S, 50S, 30S ribosome subunits, one also find a small population of 100S, 130S, 150S subunits, and EDTA dissociate these larger ribosomal subunits into 50S and 30S, suggesting that they both are subunits. Upon the addition of cations, they reassociate into 70S, but none of the other forms could be detected. There is no other forms are formed. What is the reason for not obtaining more above 70S? In this dissociation, why they are not getting 70S means that is answer is C, that 100S, 130S etc. are represent a polysome that cannot be resemble the de novo without cellular compounds. If there is if it is resemble the de novo means you will get the more than 70S form. Okay, very easiest question. A newly identified sequence are experimentally tested in vitro transport assay using the radio labeling protein containing the sequence to test import to mitochondria. Transport assay was done for a short time without membrane potential, and after the assay the mitochondria were either treated or not treated with proteinase K. Here the Main matter is the proteinase K. At the end of the assay, mitochondria were plate, platelet, pelleted, and total proteins of a pellet was isolated, separated by the STS page and autoradiograph. The representative autoradiogram is shown below. Based on this experimental data, which of the following statement is not correct? Okay, they are using this is the that samples are loaded, and digested samples are separated by the STS page. This separated STS pages are they are expressing their values. So D is the answer. The protein associated with the outer mitochondrial membrane. How they mention the outer membrane, outer mitochondrial membrane means if the membrane potential is this plus minus value, that means that is in active mode. In that it's active, another one is inactive, then active mode, another one is inactive mode. Here you can see this band. Okay, then next concept is in that way my membrane potential, that cells are in the active mode. And one more thing about uh, this activation, this protein activation is happen in the mitochondrial. How it's we can tell it's mitochondrial activation means during this separation and with separation, without separation, that will appear in the cells. So only they mention the plus value. So after that, if there is proteinase activation may happen or may not happen, there was the protein is present in the mitochondrial. So only they mention its present in the outer membrane potential. Next question, the proteosome. If a proteosome inhibitor is added, synchronously cycling human cells in G2 phase, which one of the following event is likely happen? That happen process is the arrest these cells in anaphase. Is a direct question. Just refresh the mitosis meiosis process. That is very important concept in the cellular organization process. We have a next session on the each and every topic. We will discuss elaborately in that portion. Next is the next question. A null mutation is created in a gene which is responsible for specific phosphorylation at 6th carbon position of mannose on acid hydrolase occurring cis Golgi. This following statements given towards explain the effect of mutation. This effect of mutation, they are provided the three statement, which is correct means P and Q is the correct reason for the statements combination. The combination of statements will explain effect of mutation if acid hydrolysis in the mutation not get degrade. During the acid hydrolysis, the mutant does not get degrade because that lysosome will devoid the lysosome enzyme, and that lysozyme enzymes will be segregated out. These two reasons are major reason for the non-degradation property during the acid hydrolysis. This is all about the unit 2 questions, part C, unit 2 questions. In upcoming session, we will discuss the near upcoming units. Okay, we will discuss the unit 3. We want to discuss 3, 6, 7, 8, 9, 10, 11. Okay, 11, 12 we will discuss in upcoming days. I welcome you all to next day, that is day 24 class. Now I would like to say thank you, bye. If you have any doubts, please post it in chat box, and you can